Alright everyone, I want to go through chapter 7 with you to help you prep for your test a little bit. Now, chapter 7 is pretty dense, so what I'm going to go through today are some of the high points. Hopefully it'll give you some things to think about and some places to go look up more stuff if you're not sure exactly what's happening. So we began talking about cellular respiration in the biggest sense and putting it in context with photosynthesis. So we talked about how the cellular respiration, which is the focus of this chapter, is how we transition organic molecules in O2 through a process that ultimately gives off as waste CO2 and H2O, but during this creates ATP that's going to allow us to do cellular work. The next chapter we look at is going to be the opposite of this, which is where CO2 plus H2O is going to return to organic molecules in O2. And since respiration was exergonic, photosynthesis must be endergonic, and the energy we get for photosynthesis comes from the sun. So in a full ecosystem, we take the sun's energy and turn it into organic molecules, and then all organisms, including plants, now take those organic molecules and respire them via the mitochondria to make ATP to do cellular work. So when we talk about catabolic pathways, in this case we're going to talk about how they're going to oxidize these organic fuels to make energy. So we have two processes we're going to talk about. Fermentation, that we'll cover at the end. That's a partial degradation that occurs without oxygen. And then most of the focus of this chapter is on aerobic respiration, which is where we're going to oxidize organic molecules using oxygen, or requiring oxygen. The formula for this is one I hope that you've seen many times. C6H12O6 plus 6O2 gives you 6CO2, 6HO2, and then energy, which we typically talk about as just ATP, but realize that any energy that doesn't get into ATP is going to be released as heat. So just a quick review of redox reactions. We're talking about the transfer of electrons. So oxidation, you're going to lose. Reduction, you're going to gain on oil rig. So here, this molecule X that has an extra electron is going to become oxidized when it becomes X without an electron. Y that has no electron here becomes reduced when it gains a electron on the other side. Now, one thing we talked about that's a little bit different than you might have thought about in chemistry is that redox reactions don't have to transfer electrons. They could just move electrons closer to the electronegative atom, in many cases for us oxygen, and that is also going to release energy. So here where we talk about CH4 plus O2 going to CO2 and energy in H2O, the electrons in CH4 will move away from the carbon when the molecule gains oxygen, because those electrons will go towards the electronegative oxygen molecules. So we would say that this molecule is becoming oxidized. Carbon is losing the electrons. O2, on the other hand, in this case, is going to water. O2 is becoming reduced because when we add those H's on, the electrons move closer to the oxygen. So this is a reduction and this is an oxidation. So we're going to oxidize things such as glucose, and we're going to reduce things such as O2 in cellular respiration. And you can see C6H12O6 to 6CO2, that's becoming oxidized. And one trick to see this is compare the ratios of carbons to oxygens. They're 1 to 1 here. They're 1 to 2 here. Carbon is literally oxidized. It has more oxygen. On the other hand, O2 is becoming reduced when it goes to H2O. Now, why are we doing all this oxidation? What the oxidation is going to allow us to do is it's going to allow us to reduce a molecule known as NAD+, nicotinamide. And we're going to call NAD+ an electron acceptor because it's going to take those electrons from the oxidation reaction and going to turn into the reduced form of NAD+ we call NADH. And we can think about this as stored energy. So NAD plus is a large molecule. I don't need you guys to know the structure of this. But if we add a hydrogen and we add electrons, then we are going to get an NADH. We can think about this as carrying electrons to somewhere else in the cellular respiration process. Ultimately, you guys now know that we're going to pass these through the electron transport chain. So in this electron transport chain, we're going to pass these electrons down through a series of molecules, stepwise, to release a little bit of energy at a time. So we compared this to simply combusting hydrogen here, right? So if you go H2 plus O2 to give you H2O, uncontrolled gives you an explosion, but in the end of cellular respiration, we're going to do this stepwise, little bit by little bit, electrons moving down an electron transport chain, releasing little bits of energy at a time, and this energy can then be utilized and harvested to make a lot of ATP, and we ultimately still end with the same product, which is H2O. This hopefully now makes a lot more sense to you since you've seen the electron transport chain, but remember this when we get to that part of the chapter. So to preview the three processes we're going to talk about for glycolysis, for cellular respiration, we start with glycolysis. Glycolysis is going to break the glucose down into 2 molecules of pyruvate. So 6-carbon thing going to 3-carbon thing. And then we're going to go through pyruvate oxidation and the citric acid cycle, which is going to take our 3-carbon things into 1-carbon things, which is carbon dioxide. Carbon dioxide will then leave as the most oxidized form of carbon in this cycle. And then we take all of those electron carriers we've generated in glycolysis in the citric acid cycle, and we're going to do oxidative phosphorylation in the inner membrane of the mitochondria to make a bunch of ATP. Glycolysis occurs in the cytosol. The other 2 processes occur in the mitochondria. And you can see that here. Cytosol versus the mitochondria. When we get to that oxidative phosphorylation, that will make about 90% of the ATP that we need. And so realize that these processes before don't actually make a whole lot of ATP. And all the ATP that they make are substrate level ATP instead of oxidative ATP. So oxidative phosphorylation is going to happen in that last step. Substrate level phosphorylation is going to occur earlier, and this is where we're going to transfer a phosphate group directly from a substrate to an ADP. So when we look at glycolysis, you'll think about these phosphates are coming right from sugar molecules instead of coming from free-floating inorganic phosphate. And you can see here's a substrate with a phosphate on it. An enzyme simply grabs that substrate, grabs the ATP, and moves the phosphate from here to here, giving you an unphosphorylated product and ATP. So that brings us to glycolysis. Glycolysis, splitting sugar. It's going to break glucose into 2 molecules of pyruvate and do some oxidation along the way. We talked about how it has an energy investment and an energy payoff phase. So we talked about how you have to make money, you have to spend some money to make some money. In the energy investment phase, we're going to actually use 2 ATP molecules so that we can pay off 4 ATP molecules in the end. That means we're going to only net 2 ATP molecules. As we go through the process, we're also going to make 2 NADHs that we can pass forward to oxidative phosphorylation. This process is going to occur whether or not O2 is present. And we talked about how glycolysis must be a more ancient form of the respiration process since it can occur in the absence of oxygen and it's in the cytosol. So it would have occurred in a proto-eukaryotic cell before that cell took on a mitochondria. So here it is in the cytosol, and let's look at the overall steps. So the energy investment phase, we have glucose. We're going to put in 2 ATPs. That's going to give us ADPs left over, but this is energy spent to get to the energy payoff phase, where we will make 4 ATPs and 2 NADHs and 2 pyruvates. So know your overall math here that you net 2 pyruvates, 2 ATPs, and 2 NADHs from glycolysis. Now we talked about the steps. You are not responsible for knowing all of the processes that go through glycolysis, but I would hope that if you were given a picture like this, you could sort of look at it and kind of narrate what's going on. Realize that in a step like this, energy is being used. Know that a word like kinase means an enzyme that phosphorylates. An isomerase is a rearranging, so a lot of steps are simply rearranging the molecules we have so they can be ready for the next step. Here's another kinase that's going to add a phosphate. So to notice, in investment, we're going to add these two ATPs. We're going to end up with a molecule known as G3P, glyceraldehyde 3-phosphate. That's what we will pass on to the energy payoff phase. So in the energy payoff phase, that G3P is first going to get oxidized. You can tell it's getting oxidized because we're creating an NADH. It's going to then have its phosphates pulled off, a phosphate pulled off to do some substrate level phosphorylation. That's going to go through some more rearrangement, have another phosphate pulled off for more substrate level phosphorylation, and ultimately end with pyruvate. One thing to realize here is that all of these numbers on top are 2's, because 2 glyceraldehyde 3-phosphates go through this process for every 1 glucose. Again, I'd like you to be able to look at this as a map and sort of talk about it, but only if I were to give you this picture, not to sort of memorize this in any way, shape, or form. So once we end up with that pyruvate, we can pass that pyruvate into the mitochondria to do pyruvate oxidation and then the citric acid cycle. So before we can begin the citric acid cycle, we have to get that pyruvate into an acetyl coenzyme A, which is going to link these two processes together. I tend to put pyruvate oxidation just with the citric acid cycle. So when I do the math about the process, I think about it as just being part of the citric acid cycle. So all is going to occur in the mitochondria. And so here's our pyruvate. To oxidize it, we are going to obviously generate an NADH. We will actually kick off a CO2 and then take the remaining 2 carbons, so 3 carbons to 2 carbons, 1 comes off, put the 2 carbons on coenzyme A, which is a coenzyme that can help carry these 2 carbons into the citric acid cycle. This coenzyme A is going to bring them in, drop them off into the cycle, and this cycle will go around, and as it goes around, it's going to pull off each one of those carbons individually to create 2 CO2s. It's going to do several oxidation steps to make 3 more NADHs. We will also make FADH2, which is another oxidation step, or our FAD is being reduced while the citric acid cycle is oxidizing. And we're going to make 1 ATP. So 1 pyruvate is going to make 4 NADHs, 3 plus 1. It's going to make 1 FADH2, and it's going to make 1 ATP. But then remember that 1 glucose has 2 pyruvates, so we're actually going to make 8 NADHs, 2 FADH2s, and 2 ATPs from 1 glucose through the citric acid cycle. Just like with glycolysis, we talked about the entire process. You do not need to know all of the mechanistic steps of the citric acid cycle, but given a picture like this, you should be able to recognize that step 3, for example, is an oxidative step. That isocitrate is being oxidized to ketoglutarate, and you can tell that it's oxidized because NAD+ is being reduced. You should be able to recognize that once we get down to here, we've gone from a 4 carbon plus 2 to a 6 carbon. 6 carbon gets oxidized, 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 and it returns to a 4 carbon. And everything on the backside of this cycle is simply rearranging the molecule so that you can accept these 2 carbons again. So this cycle keeps going around and around and around and around. Every glucose is going to represent 2 trips around this cycle. Alright, so once we've done glycolysis and cellular respiration, we're going to go to oxidative phosphorylation. So oxidative phosphorylation is going to be where we take advantage of all those NADHs and FADH2s we've generated. So at this point we have 10 NADHs, 2 from glycolysis and 8 from citric acid cycle. We have 2 FADH2s from the citric acid cycle. These electron carriers are going to donate electrons to the electron transport chain. So the electron transport chain is found in the inner membrane of the mitochondria, and what you can think about is that it's made up of several protein complexes. There are several subunits, each of which is an electron acceptor, and they all have increasing electronegativity. So NADH is the least electronegative, and this molecule FMN will take those electrons from NADH because it's more electronegative. This is more electronegative, more, until you get to the most electronegative, oxygen, that will take those electrons and become reduced to H2O. By going through these processes stepwise, we're going to release little bits of energy that can be utilized by the cell, and this is going to prevent us from having an explosion as we go from H plus and O2 to H2O, and more so allow us to generate ATP. You'll notice that FADH2 has less free energy relative to NADH2, and so that'll become important when we think about how we get less ATP per FADH2 than we do per NADH. Now, what is this work that's happening in the electron transport chain? The work that's going to happen is that we're going to use the electron transport chain to pump hydrogen across the membrane. So we're going to do active transport of hydrogen as these electrons move through the electron transport chain, pushing more and more hydrogen outside of the matrix of the mitochondria into the inner membrane space. Once this concentration of H plus is suitably high, it will be allowed to return through passive transport through an enzyme known as ATP synthase in a process we call chemiosmosis. So we pump out lots of H+, we allow that H+ to come through. When it comes through the ATP synthase is a membrane protein that will actually rotate as H+ goes through it, so it turns a chemical gradient into a mechanical force. As H+ has come through, they rotate this rotor. As this rotates, it's going to turn a stalk that's going to interact with the catalytic knob. We saw a video of this in class. There's a video of this in your OneNote. As this rod rotates, it's going to do allosteric regulation of the catalytic subunits, allowing them to first accept an ADP and a PI, and then lower the activation energy so that we can create an ATP by phosphorylating that ADP with the inorganic phosphate, and this will then continually spit out ATP as long as H pluses are flowing through the motor. So think about this like a water wheel, where as long as this is spinning, we're able to make ATP. This is a really cool molecule. If you look at the video, look it up online, we have a really good understanding of how this works and what this looks like inside of the cell. And so that's oxidative phosphorylation. That's the last step. That lets us take all of the NADHs and FADH2s and make anywhere from 26 to 34 ATPs, depending. There's a lot of variation in how many ATPs can be generated by oxidative phosphorylation because of efficiencies in the process. So 7.5 is going to talk about fermentation and anaerobic respiration. That's going to be when we don't have oxygen. So if we don't have oxygen, nothing that I just talked about in the electron transport chain will happen, because if you remember, we require oxygen for the electron transport chain to work. If we don't have oxygen, the electron transport chain will not work, no hydrogen will be pumped, and no ATP will be generated. So in the absence of oxygen, we're going to cut our synthesis of ATP from 32 to 2, where we're only going to carry out glycolysis. So glycolysis is going to be the only process that still occurs in the absence of oxygen, and we're going to have to use fermentation or anaerobic respiration to sort of limp along, producing as much ATP as we can. So the big problem with the lack of oxygen is that without oxygen, we are going to create a lot of NADH from glycolysis, and then we're never going to turn that NADH back into NAD+, and so we will ultimately stop glycolysis when we run out of NAD+. And so both of the processes of alcoholic fermentation and anaerobic respiration are primarily just taking NADH, and turning it back into NAD+. So here's glycolysis, going to make 2 ATPs, get us to a pyruvate, In alcoholic fermentation, we're going to rearrange that pyruvate and pull off CO2. Then we're going to take this molecule and reduce it while oxidizing NADH back to NAD+, and by returning NADH to NAD+, we can continue to do glycolysis. The byproducts of this are going to be CO2 and ethanol. Look at a very similar process of lactic acid fermentation. While this would occur in bacteria perhaps, or organisms such as yeast, when we look at when we look at lactic acid fermentation, that's going to occur in humans. So when we get our pyruvates, we're then going to turn the pyruvates into lactate. You've probably heard about this when you work out in terms of lactic acid. But the goal of moving pyruvate to lactate is that we're going to then oxidize NADH back to NAD+ and allow glycolysis to continue functioning. So as I mentioned before, anaerobic versus aerobic respiration, all of them use glycolysis to get 2 ATPs. All of them use NAD+ as an oxidizing agent, but in fermentation, we have to oxidize that NADH away from the mitochondria. We have to use pyruvate or acetaldehyde to take those electrons. In respiration, we're going to put those to the electron transport chain and ultimately to oxygen. And so if you think about the math, 2 ATPs if we're doing anaerobic respiration or fermentation, 32 to 38 if we're doing aerobic respiration. So in the absence of oxygen, pyruvate goes to processes in the cytosol. With oxygen, we're going to then go to the mitochondria and do everything else that we talked about. All right, and the last part of this chapter, 7.6, is just that glycolysis and citric acid cycle connect to all the other metabolic pathways in the cell. And we don't need to worry about all the details of this. If you take a biochemistry course in college, you'll spend an exquisite amount of time with each one of these processes. But recognize that carbohydrates aren't the only thing we eat. We also consume fats, we also consume proteins. All of these go through various processes to get broken down into different subunits, and these subunits enter into the pathway to make ATP in different places. And so if you think about entering in a different place, the numbers of ATPs you're going to get will be different for different molecules. One thing to think about is fatty acids actually enter at acetyl-CoA. And so remember, acetyl-CoA was where we took a 3-carbon thing and we ripped off 2 carbon pieces, and we put 2 carbon pieces in the citric acid cycle. If you think about a fatty acid as a long carbon chain, 10, 15, 20 carbons, you can think about how many CoAs you're going to get from one fatty acid, and you can start to realize why fatty acids are a more dense source of energy for the cell, even though they might not be as preferred as sugars. And so basically for a picture like this, I just want you to be able to look at it, think about the fact that proteins and fats also have to get into this process somewhere. Because they enter into different places, they generate different amounts of energy. That should be everything for this chapter. I know I went through it quickly, but there are a lot of notes on OneNote, and there's your textbook, and a lot of sources online that you can look at to study, but hopefully this gave you a few ideas of things to think about. Shoot me an email if you have any other questions. Good luck.